Hello and welcome to the fourth in the series of talks in which we'd like to consider the biblical account of Genesis in the light of modern science and ask the question, has a belief in the first few chapters of the book of Genesis been proven unreasonable by modern science, or can we take it at its word and still believe in 6 days of creation about 6,000 years ago? In this presentation we'll be looking at the life around us and see what it can tell us about the origins and particularly about the way the many forms of life that we see around us have come from. And we will be asking the question, is this evidence of millions of years of evolution as described by science, or does it fit the evidence that the Bible may be correct and that God made the world 6,000 years ago and life on it? So these are our choices. Is the tree of life and the gradual development of life from the simple to the complex a sensible consideration and response to the evidence we can learn from life around us? Or does the evidence support what some would say is an old and out-of-date book? We want in this series to look at the evidence so you can decide which of the two is the most reasonable position for you to take. And we'll also consider, is there a middle way? Is it possible or reasonable to hold the middle ground and accept that the world was created, but that science tells us how it developed? And this is usually called theistic evolution. So that's a big subject, and we'll never be able to look at all aspects of it, but we hope to open your minds to the possibilities. To consider the subject, we'll split it down into various parts. There have been and will be some overlap in these subjects. Until we get to the end, we won't be able to draw many conclusions, so we hope that you'll stay with us through the series. As Chris talked about in the last presentation, we looked at the record in and of the rocks and concluded that catastrophic events could have been, in fact, are so very often a better fit for geological processes that could have formed the rocks that we see today. In the second presentation, we looked at the complexity of life and how it may have started, and we concluded that just a single protein was so complex that it was unreasonable to think that it could have happened by chance. And the simple forms of life need 75 proteins amongst many other complex molecules. So the idea put forward by the commonly accepted view that life started spontaneously without a creative act is really bereft of support. We'd also like to point out, as we have done previously, that we don't have the solution to every problem, but we also respectfully claim that neither does science. And we'll be trying to be fair when we come to these problems and present the evidence so that you can come to your own conclusion. Though clearly we hope with us that you all agree that the scientific evidence makes the biblical account of creation a rational conclusion. So our consideration in this presentation, the origin and the diversity of life. We want to continue our consideration here as it builds on the conclusions from the last 3 classes and continues to evidence an alternative narrative that we started particularly strongly in the last presentation, that life on Earth is perhaps not as old as we are told it is. The current position that science takes is the life we see today is a product of slow and gradual change, powered by natural processes, that life started on Earth about 3.7 billion years ago and suddenly started to expand with the Cambrian explosion about 540 million years ago into what we might recognise today as life forms and ecosystems. We challenged the idea that the record in the rock supports the hypothesis in this last presentation, but we'll start our considerations this evening from the generally accepted premise of slow change over time. The alternative position, as we have said, is that life was created in 6 days, about 6,000 years ago. And on the face of it, these two views are not compatible. How can we take the biblical account seriously in the light of these discoveries? So let's look at the scientific position. Now, we've met these two gentlemen a few times now. As we've said before, neither was the first to propose their theories for which their names have become synonymous. But perhaps their works, more than any other, came to be the start of the move towards the currently generally accepted scientific position. Charles Lyell's book, The Principles of Geology, made the suggestion that rocks were laid down slowly over long periods of time, and that gave room for Darwin's theory of evolution as the origin of species. But it's important to know that the premise of their works, both men started with the idea that what we see around us is not the result of a creator, but of natural processes. So Darwin proposed that natural, gentle pressure of competition and chance mutations were sufficient for life to develop from the simple to the complex, Lyell having provided the time necessary for this to take place. And both men claimed that the fossil record supported their proposal. In this presentation, we want to look with an open mind at the evidence and to consider, with what we know today from modern science, that the suggestion that evolution is the source and diversity of life is still a reasonable position to hold. We are told by many, we see it on our televisions, in nature programs, it is taught in our schools, the theory of evolution is now fact. But does that fit the evidence? So let's have a look at the work of Charles Darwin. On his adventure on the Beagle and at the Galapagos Islands, Darwin observed finches on the various islands had different beak forms. These suited the particular vegetation and feeding methods of the finches on their particular island. He then postulated that all had come from a common ancestor, and the natural gentle pressure of competition was sufficient to favour the finches with beak forms that best suited the environment that they found themselves in. And so those with a more favourable beak form bred more successfully, became the dominant style of finch on that island, eventually leading to the extinction of other finches with less well-suited beaks. But he went on to suggest that if natural forces could lead to the development of a new beak form, then given enough time, the same natural processes could lead to completely different life forms. And then that all life on Earth had a common ancestor. That, life having started, though he knew not how, could have developed from, as he saw it, simple single-celled creatures to the life we see today, and even ourselves. As we have said, this was not the first time that this theory had been proposed, but Darwin's theory of evolution as the origin of species was very appealing. For the first time, it laid out a story of how this could have happened. But in the intervening years, science has moved on. For example, we have considered previously a single-celled animal is now known to be anything but simple at all. But just for a moment, let's consider the story and the development of his theory. Now, Darwin himself is often considered to be a scientist, but actually his method was not very scientific at all. His theory is better described as a philosophy, because it's an idea that he proposed. Now, it's quite reasonable in scientific method to propose an idea, but this then must be backed up by experiment and evidence. Now, Darwin gained his degree in theology, ironically the study of the Bible. So he was never trained in the scientific method. So he did no experiments or field work to back up his theory. Rather, he relied on a good story, an extrapolation, which is not evidence in support of his theory, but just what-if conjecture. In any other field of science, philosophy would need an enormous amount of proof and consideration before it could be taken forward. But in evolution, it seems that a good story is just enough. In the second presentation, we looked at the origins of life, and we showed that a simple protein was actually fantastically complicated, far too complicated to have happened by chance. But rather, proteins are made in the cells of our body from instructions in our DNA. We determined that there is no current plausible scientific suggestion for how life could really have started. So let's look at some modern science and see what it tells us. As we said previously, it wasn't until the 1940s and the 1950s that experiments pointed to DNA and the now commonly known double helix pattern of DNA was shown to us by Crick and Watson in 1953. In 2003, the Human Genome Project was declared complete when one complete human genome was sequenced, and since then many have been. But this all started 130 years after Darwin. He knew nothing at all of any of this. We now know that to develop a new feature is all about information. The instructions to make and operate every living thing is stored in its DNA. And to make a new feature or a new protein or anything within our bodies, we need new DNA. And DNA is the most complicated and compact information storage system known to man anywhere. So let's consider something that we're perhaps a bit more familiar with, computer programs. Now, it's quite like a DNA in that both are instructions to do something. Now, if you and I went into a computer program and made a few changes, not knowing at all what we were doing, well, what would be the outcome? Will the program work better? Well, we know it probably won't. And this is the crux of the problem: information. The fact is, there is only one observable source of information anywhere at any time, and that is that information comes from intelligence. And we shall see as we go that natural random processes have quite a different outcome when they are allowed to act on information than what we would expect. So let's have a look at what is happening to life today, as current experimental data shows us is the case. This is not speculation, it is evidenced. We'll take these as a series of challenges and see what we can learn from them, examining them one at a time. So the first one will be: most mutations are slightly deleterious. To stop these passing forward, we need the creature to die before it can breed. Advantageous mutations are rare and offer usually only a small advantageous, too small to be selected for. And advantageous mutations do not build up to new features. So the first one, mutations are slightly deleterious. So just as with a computer code, as we thought about, if we change a letter or number randomly, the likelihood is the program will not work as well as it did. We know this by instinct, and it's the same for a DNA. But there are other ways of looking at this in a slightly more scientific way. The second law of thermodynamics is worded in many ways. I took this one from Wikipedia. In all natural processes that occur, including spontaneous processes, the total entropy of a system and its surroundings increase, and the process is irreversible in a thermodynamic sense. The increase in entropy accounts for the irreversibility of natural processes and the asymmetry between the future and the past. Oh, that was a lot of the technical wording, wasn't it? So, second law of thermodynamics, what is that about? Well, actually, we can show it in a more simple way, and we do really understand it already. Take a cup of tea. When it's just poured, it's hot, but soon it goes cold. So where's that heat gone? It hasn't just disappeared. Your room has got a little warmer. So the total energy, the high energy that was in the tea, has found a new lower level, has shared its energy with the room, and your room has got slightly warmer, so that eventually that both are the same temperature. So there is no known exception to this law. Entropy is the lack of order or the unavailability of energy to do work. The cold cup of tea can no longer make you warm. And everything that has energy or a state of order higher than the surrounding will, over time, find a new lower level until everything shares the same lower level. And this is an increase in entropy. It accounts for everything, not just cups of tea, shiny cars and our drives. We'll eventually just turn back to the elements which they came from and rust away to nothing. So, there are no known exceptions to this law, while apparently except evolution, in which it is proposed that order and complexity increases. So, entropy reduces. This is in violation of that fundamental law of our universe. The conclusion of this is that biological information is inherently subject also to entropy and decay. This underlines and supports what we already know. If you mess with the computer code, it's going to get worse. And if you keep messing with it, eventually it will stop working altogether. And so it is with DNA. Random changes in the code will slowly result in degrading of the instructions in the code. There will be occasional lucky occurrences by chance when a better alternative is found, one that works or one that indeed may be better than the original one, but these are far and few between. Remember back in the second presentation how critical it was to get the amino acids in a protein in just the right order. A few changes to that string of amino acids and they won't fold properly into a protein, and then the protein won't be able to do its job, leading to a reduction in functionality, reduction in order, and an increase in entropy. You may remember the enormous odds that are required to make a protein by chance, and these are the sorts of odds that we're looking at, finding mutations by chance that work better than the current ones we have. So let's look at what is needed to happen to stop these deleterious mutations passing forward, which of course is needed for evolution to work. This has been considered many times by many top scientists, right from the early days of the study of population genetics. It's become known as Haldane's dilemma. Now, Haldane was a Darwinian scientist. That is, he believed in Darwin's theory of evolution. In fact, he was the founder of what is currently taken as the amended version of Darwin's theory. But he realized also that there was a problem. Bad mutations were more likely than good ones. And to remove the bad mutations, a mammal, in a mammalian population, the offspring must die before they can reproduce. So each pair of parents would need a larger number of children than was needed just to maintain the population, be able to breed out these bad mutations. And Haldane realized that the number of offspring that were required was too big to be reasonable. The ramifications were that populations were too small and they bred too slowly. Now Muller realized that bad mutations work like a ratchet. The term Muller's ratchet was coined way after his work, but it's a good summary of what his work came to summarize. He realized that good mutations would not come along often enough to make up for the bad ones building up. And he estimated that about half a mutation per generation was the biggest load a population could endure. More than that, evolution would go into reverse. His work was done about the time when nuclear testing was being done in the open. And radiation is a very bad thing and damages our DNA considerably. Kumara picked up on this work, and he showed that Muller was a bit optimistic with his estimation of only one mutation per generation. He estimated that two mutations were happening, and he found that at this rate, he calculated that a pair of parents would need 3.27 billion offsprings to breed out the bad mutation. Well, clearly, that's ridiculous. There's a big problem here. But with the passage of time, more have considered the problem. But unfortunately, it has not gone away. This table shows what Nachman and Crowell considered the numbers were. The mutation rate of 0.1 is 1 mutation per 10 generations. And that's just about okay. 9% of children have to die before they breed, and every adult would need 2.2 offspring to maintain a population. But by 1 mutation, we need 5.4 offspring to 14.8. And by 3 mutations per generation, it's over 40 offspring per adult. So we can see, as the number of mutations increase, the problem gets bigger very quickly. And what is the current measured rate of mutations? Well, it's about 100 per generation per person. Now, we need to comment on this before we can make any conclusions. But we can see that it's going the wrong way very quickly. This work is right up to date. And you can see that it is based on 100 new mutations per generation, per parent. And at the bottom, you'll see that the number of offspring is 10 to the power of 35, which is 10 with 35 noughts after it. And the author wrote after that it was a bonkers problem, and it certainly is bonkers. Now, there are two assumptions that all these scientists have been making. Firstly, that our genome is mostly junk. Originally, it was assumed that only 10% of our DNA was functional. The rest was junk left over from our evolutionary past. Well, we now know that it's about 80% functional, or perhaps it might be better to say we know what 80% of it does. The remainder is a mystery to us at the present. So this makes comparisons between the different informations that we've been looking at difficult to make, because the early ones were assuming that of the 100 mutations, only a few would fall in parts of the code which were functional. The second problem is that all these scientists are assuming a long lifespan of the human population of hundreds of thousands of years, as is assumed by evolution, but it's not the case in the biblical account. But the fact remains that it's understood that however you look at it, there's a problem. And we'll come back to this towards the end of the presentation. Let's have a look at it from another way. Let's think of something that breeds much more quickly, because that's one way out of the problem as we've been looking at. Let's take the flu virus. It's a topical viruses at the moment. The Spanish flu killed about 50 million people in 1918 to 20, which is an enormous number, more than that died in the war. But today, the flu virus that killed so many people has gone, and human H1N1 is thought to have become extinct in about 2009. Now, we know that viruses mutate, and we understand them to build immunity to treatment. It's called antibiotic resistance. But what really is happening in this process? Well, an antibiotic usually recognizes a particular protein within the virus, and it will latch onto that and eventually destroy that virus through the gateway that's presented by the particular protein. So if a part of the flu virus doesn't have that protein, then the antibiotic can't work on it, and that is how it builds resistance. But you'll understand then that a virus is losing proteins all the time, not gaining new information or gaining resistance, but losing information to gain immunity. And it carries on doing this until it can't function anymore. And just like the flu virus, it eventually disappears. So, the last two challenges in our list are about advantageous mutations. And clearly, these must be effective if evolution is to work. So advantageous mutations are rare, we are suggesting, and only generally offer a small advantage, too small to be selected for. And advantageous mutations don't build up to new features because mutations are blind, they have no direction. Advantageous mutations are not sufficient to outbalance the deleterious mutations because they come along so rarely. Many mutations are required to swap out just one amino acid in a protein. You remember from that study we looked at a few presentations ago, how the DNA instruction is brought forward to make a protein. And many instructions within the DNA are required to swap out and to change the function of a protein. And they can't build up to a new feature when we think about it at this level, because these small changes are, well, just small changes. If we need many changes to swap out one amino acid, just imagine how many changes are required to change an arm, say, into a wing. And the process is blind. It is random changes. It can't plan or make a direction. So it's quite reasonable, just by our own understanding of the thought of the process, that advantageous mutations will not build up to a completely new feature. So let's look at some of the problems with making evolution work. Let's now consider what we do know. And again, we'll consider these points in a bit more detail as we go. Now, the first one of these points we've already looked at. Information comes from intelligence. In fact, it is the only observed source of information is intelligence. Information doesn't come from random processes. So what can natural selection do? What is happening in the finches that Darwin observed on the Galapagos Islands in their beaks? Well, it's clear that more successful creatures will breed more successfully than less successful ones. That's no surprise to us at all. And adaptation is fine. It's fine-tuning of a particular creature to suit its environment, so that in this case the finch can remain a finch and be successful within its environment. And this is observable science. We know it happens. It's built in to our structure and our DNA. But there is no evidence for the development of life from one life form to another in the fossil record. Adaptation doesn't lead to new life forms, as we have looked at previously. In fact, this is a quote from Darwin's own writings in The Origin of Species in Chapter 9. Why then is it not every globe geological formation in every stratum full of such intermediate links? Geology assuredly does not reveal such finely graduated organic chain. And this, perhaps, is the most obvious and serious objection that can be argued against his theory. But he didn't give up. He thought that the explanation lies in the extreme imperfections of the geological record. He was expecting that as we found more fossils, eventually we would find the missing links. But 150 years after his work, and with about 250 million catalogued fossils and too many others to count, a few in my shed, there is still no evidence for this gradual change. Even current enthusiastic proponents of the theory of evolution acknowledge this problem. Stephen Jay Gould wrote this: "The extreme rarity of transvisual forms in the fossil record persists as the trade secret of paleontology. The evolutionary trees that adorn our textbooks have data at their tips and nodes of their branches. The rest is inference, however reasonable, not evidence of the fossils." Eldridge and Tattersall wrote, 120 years after the paleontological research later, it has become abundantly clear that the fossil record will not confirm this part of Darwin's predictions. So, the fossil record shows us that creatures appeared. They remained largely unchanged, apart from small adaptations, and then disappear. If we were to represent this in time, then it would look not like a tree, but a series of sticks parallel to each other, not joining. And even Stephen Jay Gould had to admit that, as we've talked about in earlier presentations, there is no support for a tree-like structure of development of life forms. In fact, it's pretty much the opposite. Life was much more diverse in the past. From a previous class, we remember that there were 20 classes of arthropods described from the Burgess shales when arthropods first appeared, but there are only 5 classes today: crabs and lobsters, spiders and scorpions, centipedes, millipedes and insects. So there are less now than there were right at the beginning. Now, life was more diverse in the past. There are many forms of life that we see in the fossil record that are not with us today, and indeed many different forms of humans as well. And this can be a challenge for creationists, and if it is a particular problem for anybody, please check the reference list at the end of this presentation, as we don't really have time to go through it today. But it is also evident and topical that as a race, we still don't like diversity. As we draw this presentation to an end, let's just come back to the problem of deleterious mutations. This has a serious implication for our series, our species as human beings, and life on our planet. We've looked at genetic information is not increasing, it is actually reducing. Here is a quote from Lynch. The increase of the variety of afflictions, including autism, male infertility, asthma, immune system disorders, diabetes, etc., already exhibit increases exceeding the expected rate. The long-term consequences of such effects is a genetic deterioration in the baseline human condition. And we could add to this list dementia and Alzheimer's disease, and Parkinson's disease is also understood to be linked to problems within mitochondrial DNA. Yet from the biblical perspective, we know that people lived a lot longer in the past. We can see around us in the populations of our relatives, problems of genetic deterioration increasing all the time, with the health of our groups of people deteriorating. We'll need to come back to this in the last presentation, but it is evident from life on Earth is not continuing to develop, but is going the other way. Natural forces are not the driver to develop life, but are degrading it at an alarming rate. Humanity is a dying race on a dying planet. So science has given us a good story, that life started simply and developed by natural forces, but we have found this is not really supported by the evidence. So have we proved that God made everything in 6 days? Well, I don't think so necessarily. But in the first presentation, we chipped away at that foundation of the bedstone of science, the scientific position that the world is a very old place. We found that there are no natural processes that come close to the explanation of how origin and the complexity of the simplest life forms started. In the third presentation, we showed that catastrophic events were a better model for the formation of geological forms that we see around us today than the slow processes we are told happened. In this presentation, we have seen far from developing from the simple to the complex. Life started complex in the beginning, and diversity has only been reducing since. The evidence, if it's left to itself, life is not increasing, but is on the way out. We're not developing higher life forms. So, in this the last presentation, we'll pull all this together and try and build a new picture of what is really happening, what the ramifications are for humanity, and particularly what they are for ourselves. So we hope that you'll join us again. As always, we provide a list of references. And that second one, Todd Wood, 8 Men to Adam, is the one I referred to if you'd like to look that up if you have an interest in different forms of human life before the present. I hope you've found what we've discussed this evening to be useful.